And for this last session of the year, it's my pleasure to receive Dr. Sabrina Amador-Vargas, who is a researcher at the Smithsonian Tropical Research Institute in Panama and at the head of the Tropical Behavioral Ecology Lab. So in Dr. Amador-Vargas' lab, they are studying mutualism of ants with the swollen-thorn acacias to better understand behavioral, individual or social or morphological modifications. Today, Dr. Amador-Vargas is going to explore the role of fungal rhizomorph in bird nest as ant deterrents. Dr. Amador-Vargas, the floor is yours.
Thank you so much. Thanks for joining. So it is my pleasure to introduce a little bit of the research that has to do more with the interactions and a lot of organisms interacting around like its characteristics in the tropics. So when we talked about ecological interactions, we can think on events that happen during one particular time, like predation, or mutualisms. That those mutualisms could be sometimes obligate associations in their more extreme form. For example, the wasps and the figs, in which one depends entirely on the other. And the same thing happens with the ants and the acacia ants, in which a plant is defended by a colony of ants, and their fitness depends on having the other organism present. These obligate mutualisms many times function like a novel environment where other organisms can develop adaptations and strategies. And besides that, we also know that social insects and social insect colonies are also promoters of specialization. And I have two very cool examples here of beetles that resemble the ants where they camouflage and live with. And another example more recent of beetles, again, mimicking termites to live inside their nests. So just by being a social group and a very abundant and in common in the environment, they have pushed other organisms to develop extreme adaptations. And on top of that, when these social insects are associated with other plants or fungi, that also triggers the specialization of organisms. For those of you who are not familiar with the swollen-thorn acacias, those are the pioneer trees, not too big. So in that picture, you're looking at a huge fig tree, and in front of it, the acacia tree that I am showing now in color, so it's easier for you to see. So it's not that tall of a tree, at least the species that I work with, and not very thick. It will live for 2 decades maybe, and gets replaced by older forests. Here's another example of another picture of a young swollen-thorn acacias. They evolved from a large group of species in the genus Bachellia, no longer called acacia, but I use acacia as a common name. And from that group, at least 13 species of plants evolved these particular adaptations. Domatia to host the ants, nectaries or extrafloral nectaries to feed the adults, and the American swollen-thorn acacias also evolved something called Belgian bodies, which are packages of protein. On the side of the ants, we have they belong to the genus Eudomyrmex, and this is a very, very numerous genera. And from that group, 13 species evolved associations with plants. And by looking at the names of the species, and some of the species, you start to wonder why the plants associated with these groups of Pseudomyrmex. The pseudomyrmex living in the swollen thorn acacias use the thorns as nesting spaces where they keep the food, as you can see here, and the larvae are the brood in general, and the adults also get inside the thorns to take care of the larvae, but also when it's raining and at night. I'm showing in that slide the contents of what you can see in a single thorn, and that's like repeated in the different branches of the tree. In exchange, the ants will protect the tree against potential enemies. And in that short video, you can see how they react when we mark the trees. So they're very responsive to foreign material, not only potential herbivores, but anything that they do not recognize as their host tree or part of the colony, they will attack. They attack by biting, and they have a powerful sting with painful venom. Very painful, my students know that. And they also remove debris, and using their mandibles, they try to keep their tree clean from any foreign material. And they even attack neighboring vegetation by pruning, and they use their mandibles to prune that vegetation. And so here in this picture, you're looking at this vine that was naturally growing around a sapling that I had as an experiment next to a larger tree on this side. And you can see a tiny ants crawling on the vine and cutting it with their mandibles until the vine collapses. That creates very large clearings around the tree. So the point that I'm trying to get at is that they are obsessed about having their acacia in wonderful conditions. And these trees that are defended by ants then are attractive to other organisms, and among some of those we have identified, or in different researchers have identified, roaches, birds, caterpillars, and galls nesting or raising their young in the swollen thorn acacias. And sometimes we also see organisms where the adults are also spending time there, or a large part of their time. And for example, these true bugs, they do the entire life cycle on the swollen thorn acacias. And to give you a better idea of how they look like, this is the egg case of one of these roaches. This is a humongous caterpillar that goes around feeding from the plant and not getting attacked by the ants. And also here are the spiders that can also live on the acacia. These true bugs that I was telling you about, also feeding from the plant sap, and well, sometimes they get detected by the ants. And here, just very simple to show you that the bugs are not just using the plant, because when the plant doesn't have ants, they are not there. And most of the trees with ants will have bugs, and they have a strong preference for particular species of ants. And in that case, we also tried to understand how is it that the true bugs can be around the ants. And one of the techniques they use is a strategy of lifting the legs. So when the ant walks through the main mid vein of the leaf, as it is walking, the bug is lifting the legs. So the ant is less likely to touch it with the antennae than to smell there's something different. So another picture of a true bug lifting the legs. And sometimes that works, but sometimes it doesn't. And so there is also evidence in the literature that they have some chemical strategies to deal with the ants. But now to the topic that we wanted to talk today. It's mostly the birds, those birds that like to nest on the swollen thorn acacias. And with the help of my student Catherine Parras, we studied some of the swollen thorn acacias and revisited this whole idea of why birds would use the acacias. And in that article that I don't have time to talk about today, but that you can read, we found that in the forest, the birds really, really like nesting on swollen thorn acacias. But as you move to more disturbed habitats, and more urban habitats, they lose their preference for swollen thorn acacias and they shift to nesting in very large trees in those cases. And potentially the explanation is that in these other environments or habitats, they have different enemies than what they have in the forest. And the stars of the project, in that case, just to give you an idea, it's diverse, the group of birds that like to nest on swollen thorn acacias, but they're kind of aggregated in three groups. Tyrannids, or the group of the flycatchers or kiskadis, and wrens, it's another group that uses the trees, and orioles that you're gonna see in the next slide. The shape of the nest is very different among those families, and the materials they use are also different. So this some of them will put the nest intertwined in the branches of the acacia, while others will build something that's more pensile or hanging at like the drop life like nest. And here's another flycatcher that does the same thing in a smaller scale, like a hanging nest on the tips of the branches. And what it's this bird and this other bird have in common is that most of the nest material is made of those very dark strands. So if we look closer to it, it looks like hair. And so these are nesting material, and on this side, that's a horse hair. So you can see that's the reason why the people in the communities talked about those nests are made of horse hair. And actually it's not hair, it's a fungus. And the type of growth is called a rhizomorph. And so these are organized hyphae that acquires that shape. And not surprisingly, they are called horsehair fungus. And in a trunk, they will look something like that. And that's the place where the birds will pick them up to weave their nests. This is an electron photograph that shows the structure and the external structure of the hyphae. And we also have confocal images of transversal cuts of the strands. So you can see all those tiny circles are the hyphae, which are aggregated. So they have a drier or harder layer outside, and the hyphae organizes inside. And they belong to different genus, and they're very hard to tell apart just by looking at the strands. So that's why we should call them just rhizomorphs or marasmioid rhizomorphs, because they belong to a Marasmiaceae family. So we have strands from the Marasmius genus. There's another Clinipellis, and there's another one, Gymnopus. And as you can see, just by looking at their internal structures, you can tell the genus. which is something we discovered and is very useful because without this, the only way to tell them apart is by doing barcoding or DNA. So this is cheaper. And so the story of birds and rhizomorphs goes beyond the acacias. About 176 species of birds that belong to 37 families and build their nests, putting them in different layers of the nest. The lining, the structural part, or the attachments to the branches. It varies. And the hypothesis that people have proposed for why would they incorporate that material first has to do with the antibacterial and antifungal properties of the rhizomorphs, but also the material is very strong, so it has a high tensile strength and certain repellence to water. So that was another or is another hypothesis of why many birds use these materials. And we're proposing this third one that has to do with ant deterrents. And the reason is because, as you saw before, at least this yellow olive flycatcher nests more than 90% of the time on swollen thorn acacias. And you already saw how aggressive the ants are. So the ants are walking on top of that branch, and somehow the bird is able to nest with them, and the fledglings are not attacked by the ants. And so with in a collaboration with my student Raiza Cortez, we were testing this hypothesis in Costa Rica in a national park called Palo Verde. And so what she did, the experiment that she did was testing the reaction of the ants towards the fungus fibers from vegetation. And because when the birds are nesting is during the months of higher rain, she also tried with the fungus, but this time wet. And we worked with, well, she did a lot of this work with the 25 colonies and more than 500 ants. And the fungus we later knew, or using barcoding, we knew that it belongs to marasmis. And this is an example of how the experiment looks like. And she was watching these videos frame by frame to record the behaviors of the first ants that were finding the rhizomorph strand. And yeah, annotating the interaction with the strand. The pseudomyrmex ants are very solitary, so they make a lot of decisions on their own, unlike other ants. So the behaviors, we classified them in 3 groups. We were interested in understanding what happened when they first encountered the strand, and that's classified in whether the ants react to the strand versus ignoring or walking on top of it as if it wasn't there. And sometimes their first reaction is biting, and sometimes it's just staying there and using their antennae repeatedly. After they do either of those behaviors, then they can do some other things, and we divided that in behaviors that indicate repellence. For example, backing off after contacting, maybe could be biting and then backing off, or repeating antennation and backing off. And sometimes they groom, and sometimes they turn around instead of continuing walking on that branch. And then we had to do a list of behaviors that were commonly observed, and that I will tell you more, but that initially we didn't thought we would see. For example, biting a nestmate, curling the gaster, walking in circles, and something that we call like dropwire walk, which is not like walking in circles is almost spinning on the same place, but dropwire walk is going like changing directions all the time and speeding up. Sometimes they do that, and sometimes after contacting the materials, they hide inside the first spine they have, or the spine they have closer. In general, I think I'm not showing all of it because it's too long, but I'm showing a few of the indicators of repellence, like backing off, grooming, or turning around. It was more here I'm showing the probability of observing the behavior and the with the rhizomorphs or the plant material, which is the first column, and you'll see at least for grooming and turning around, these two behaviors have a higher probability of happening after contacting the fungus than the plant. Backing off is not. Is equally likely to happen. And these other behaviors that I was telling you about were almost exclusively observed with the fungus. As you can see here again, the arrangement is these two at the end are showing the fungus, and the first one is showing whether the behavior happened with the plant fiber. And for some of them, the behavior was very, very rare with the plant or never observed with the plant. And it was happening sometimes with low probability, sometimes with high probability with the fungus. And so we think from that project, we learned that the reactions require contact from the ant. So it indicates that whatever is changing the behavior of the ants is a non-volatile, or is a chemical they need to perceive by contact with chemoreceptors, by contact chemoreceptors. The second thing that we learned is that some of those behaviors that we observe happening almost exclusively with the fungus indicate intoxication, alarm, and cognitive impairment. And some others are very hard to interpret. For example, what's the meaning of the ant curling the gaster? In previous research, we have learned that curling the gaster is something they do when they are not alarmed, so it's not showing the same, but it's hard to interpret. And then the third lesson is that because what they do most when they interact with the fungus is grooming, maybe is this constant grooming what impedes the ant from removing or cutting the material as they do with many other materials. And so this could be, in the end, the mechanism that keeps the ants away from the rhizomorphs that are being placed on the tree. And some other lessons has to do with whether the behavior evolved, the behavior from the birds evolved to deter acacians. I don't think so, because other birds use rhizomorphs when they are nesting in other types of trees, non-myrmecophytes. And we still don't know whether the rhizomorph affects other hymenopterans. And so that was the next question we aim to address, whether the rhizomorphs are also affecting the behavior of other organisms. But before I jump into that, there is a tiny detail that is very important, is that the active substances that the fungus potentially have should or require that the fungus is alive while it is part of the bird nest. And as I told you, they collect the rhizomorphs when they are alive, emerging from tree trunks or leaves. But there was discussion at the time in the literature of whether the fungus was alive in the nest or it dies. And it might be viable at the beginning of the nesting or the breeding season, but it might die afterward. And the only literature we could find about them is that for the streakback oriole, the information was at the moment that the fungus was not viable when it is on the nest, that it is dead. But more recently, there are other reports in Guyana, I think, in rainforest. The bird species was not recorded or not reported in the paper, but they do mention that the fungus is metabolically active while on the bird nest, and it was part of both the lining and the attachment layers of the nest. We weren't sure about the fungus in Costa Rica or Panama, so we first tried to understand whether the rhizomorphs are alive in the bird nests. And two ways of knowing this is through looking at the rhizomorphs and whether they produce fruiting bodies while being part of a bird nest. And also you can try to grow them and to check their viability. To do this, because I work with ants and not with fungi, recruited mycologists like the student Joel Trejos and the technician Javier Ballesteros from another lab, a lab working with fungus here. Don Jorge Ceballos, who supported us with the microscopy part, and he was key in discovering something very important. And Alice Bosser, who was doing her master's thesis with another part of the study, but she tried to tackle this question. And so Don Jorge was essential because when we were doing the images of the rhizomorphs, he was the one who told us those things are alive, I think, because I tried to rehydrate it and they are growing. And at the moment, I was thinking that they were dead. And then this was a discovery from Joel. Joel showed me the nests with the fruiting bodies. So now we have confirmation that the fungus is still alive and remains alive in the bird nest. And then with Javier and Alice, they did this viability experiments, and here we're showing you that from something that looked very dried, and we rehydrated, and you can see the hyphae growing. What's more surprising is that these nests were collected in June 2023, and we were like doing these viability tests almost a year afterwards, and those things were alive. So those nests had been sitting in my lab for almost a year while I was thinking the fungus was dead, and it's very, very alive. That means that it could produce the active substances then. And because we have the samples, we also, with a collaboration with Dr. Christine Santos at STRI, or the institution, with this bird in particular, Tolmomias, the yellow olive flycatcher, we try to understand which rhizomorphs were they using. It seems like it's Marasmius and Gymnopus. And some of the sequences that we were able to identify to species agree with the species that was recorded before, Marasmius neocliniseci. Apparently it is from the neotropics. And going back to the substances, so finding what is it that the fungus has that alters the behavior of the ants. For that, I collaborated with at the moment doctoral student Alisa Gaisi and the postdoctoral fellow Callum Kimball, and with their guidance and help, again, my two students, Joel, Raisa, and me, embarked into this trip of understanding what was in the fungus that altered the behavior of the ants. So I'm gonna go through this a little bit quickly. It's simple in principle. If you do washes of the rhizomorph in these three solvents, they are picking the substances from the most polar, the methanol, to a less polar or non-polar, the hexane is collecting those extracts. And from there, you can start to understand whether the active substances are polar or non-polar. And to do that, we did all these extracts with methanol and in filter papers, the extract, the methanol, the solvent itself, and the control. And same thing for the other two solvents that are extracting non-polar substances. And we offered this to the ants and to see what was whether we could recover some of the behaviors we had seen in the ants happening with now with just the extracts on the filter paper. So you see that ant is grooming after contacting the material and grooming a lot and keeps grooming. And so we're comparing the probability of the behaviors happening with the methanol extract with just the paper and the solvent that we evaporated the solvent. So here is an example of how the ants behave with the control. You see they're obsessed about cleaning the acacia. No grooming afterwards, or at least not immediately. So those were the videos, so you have an idea of what we were doing. And 23 colonies, 100, almost 140 ants that we tested, and we were looking to whether the ants were removing the material from the leaf, whether they groom during the interaction, meaning that they picked up the paper and release it, or whether they groom after they had discarded the piece of paper. And like that. And so the graphs that I'm showing with some of the results, again, show you the probability of this behavior happening, between the control, that is just the filter paper, the fungus extract, and the solvent. And for the methanol, the hexane, and the chloromethane, exactly the same thing. But so you don't get confused, I'm going to explain this one. So, for example, the ants were more likely to remove the paper, the filter paper with the fungus extract than any of the other two controls. They were less likely to groom during the interaction or the time they have the paper on the leaf, but they are almost always grooming after interacting with that. And they don't do that when they encounter filter papers with just the solvent or just the filter paper. For the other two solvents, there was no difference in any of the behaviors. So this is telling us that whatever substance is altering the behavior of the ants, it's on the polar part of the extract, whatever is the methanol is extracting. The people who know about this, they told me that this is what you can find in that chemical profile, and this is a compound of interest, and that was my face when I looked at that name. And when we looked at where else can we find this substance, it's also found in cordyceps and ganoderma, which is used in medicine, has medicinal uses, and parasites. And it's used for antiparasitic activity, and the reason why the fungus might have that substance is because it degrades lining, and in the end, that's what it's like the metabolic function for the fungus is probably that one. As a side effect, this substance causes all these effects on the ant, or at least that's what we know so far. And as I said, these birds using rhizomorphs occur in places where the acacia ants do not occur. So this is almost the overlaps entirely with the distribution of the swollen-thorn acacias. We can find them also here. And this is the oriole. And the yellow belly flycatcher happens in Central America, but also in South America. And the swollen-thorn acacias are not happening here. And very interestingly, the yellow belly flycatcher in South America really, really likes to nest near wasps' nests. And again, is one of the few bird species besides the oriole that loves to build the entire nest out of rhizomorphs. In most of the bird species using rhizomorphs, it's just that they add some strands. So here's an example from Panama of cacique in the same family of the oriole. And you see a wasp nest, ant nest, Azteca ant nests, and the hanging nest of the oriole. So in the work that I did with Risa, or in the collaboration with Risa and Joel, we have tested the ants in this subfamily, and you're looking at a phylogeny of ants, Pseudomyrmecini. And with Alice, we tested nearby ants in nearby species, Dolichoerine and Ectatomini. And so Alice worked with particularly Azteca ants and Ectatoma ants. And she was looking at the behavior of those known acacia ants and the rhizomorphs. And she did trials with 3 treatments, the plant, the rhizomorph, and also sowing thread. And the species Ectatoma rhythm, and the way she forced the ants into interacting with the fungus is by making a little ball and stuck it in the entrance of the nest, and that forced the ants to interact with the material. And Azteca alfari is it nests with the Cecropia plants, and so just putting the rhizomorphs on the leaves, they will eventually come and interact with it. Here's a short video of what ectatomas do. And so you can see they grab it with the mandibles and move them away from the nest entrance, and sometimes very far away. So it's no longer a problem to enter the nest now, but the ant continues moving in. And it's gonna drop it there and go back to the nest. And from those videos, Alice was able to check whether these other behaviors, whether they were grooming or doing something odd. And this is the Aztecans interacting with the material. They're smaller than Ectatoma, so some of the behaviors she couldn't record in so much detail. But still you can see whether they're biting or grooming. It's still possible to see. And this is what we were expecting. If the fungus is deterrent to these ants, they should do more ignoring of the fungus and less of the other 2 treatments, and less biting, more grooming with the fungus and less carrying, because in theory they shouldn't want to touch that material. And her results are very easy to explain because Ectatoma and Azteca, in most of the behaviors she checked, they show absolutely no differences in the probability of doing the behaviors among the 3 treatments. So they ignore or react with the opposite, more or less with the same probability. And they bite and groom with more or less the same probability in Ectatoma and Azteca. And something where some of the behaviors where she found a difference is that Ectatoma and Azteca, they both, and you're looking at the rhizomorph treatment here in the middle. The blue is the sewing thread, and the first one is the plant fiber. They carry that material, the rhizomorph. They carry it for a longer time or for farther from the nest entrance. And in summary, in the end, was the rhizomorph toxic or repellent for Ectatoma and Azteca? Apparently no, not for these 2 species. Are the reactions specific to Pseudomyrmex spinicola? Maybe, but I still doubt it. Because by looking at the videos and the behaviors of these 2 antispecies, you can easily observe that they both manipulate the material for very short time. The acacians, they bite, and because they're trying to cut the material, they interact with the material for a long time. And that's the other big difference. Pseudomyrmex is attempting to cut, and using the mandibles are really, really making a lot of force to bite. But Ectatoma and Azteca use their mandibles for holding the material and carrying away from the nest, never really chew on it. And so no, the ectatomas were not hallucinating, and it might be an effect of how they behave with the, or how do they manipulate the material. So it was a very, very interesting trip with this fungus. We have these observations, field observations of organisms, and with 1 species, we found differences. We look for 2 more species, we didn't find any differences. And so we're going back to the field to test the interactions and presenting the papers to Ectatoma and Azteca. So the substances extracted by the methanol are present on the surface of the material they're testing. And yeah, I'm running out of time, so I'm giving my very huge thanks to all the people that has been involved in this material, people who donated nests or in this project, then people at the national parks and the reserves, and millions of ants that behave very well during my experiment. And with that, I'm happy to take questions. I'm just gonna turn my lights on for a second.
Yeah, no worries. Go ahead.
I'm back.
Thanks a lot, Sabrina, for the very nice talk. Very happy that the lockdown is finished and that we were able to actually see your presentation. So we already received many questions, so like I will just start and go through it. So we are Bastor Manta Forever. So first question, that is, when the ants bite the plastic that was on the trees, do you know if that impact the health?
I guess they're talking about
It was the very beginning of the talk.
the flagging tapes.
Yes, yes.
Whether they are affected in their behavior is the question.
On the health, like if, for instance, like some, I don't know, like maybe microplastic exposure or things like this.
All right. No, so I don't have an answer for that because we haven't checked what happens to the flagging or
Okay.
the behavior of the ants with the flagging. But what I can tell you is that that's something they can easily cut, and in the end they cut. Um, Putting flagging on swollen torn acacias is not something that I do. It's something that I teach my students not to do, and usually so that video was one of those times where one of my students marked the tree with flagging.
Okay.
And, but yeah, it tells you that they are very persistent in trying to cut something, and so that's why we use metallic tags.
Okay. Thanks. So there are other question of Bastormanta, but maybe I will come to it a bit after because I would like to try to at least ask one question per person. So we have Taun with first saying that fungi are really crazy, fascinating. What's making the ants bite their nest mate after contacting the rhizomorphs?
Yeah, that was very surprising. And well, you saw the list of all those other very odd behaviors that we weren't expecting to see. Um, it seems like they get after chewing on the rhizomorph, they get more aggressive or alarmed. So it's just this the pseudomimics are very visual ants, unlike other ants. The movement of the other nestmate, it seems to trigger this reaction of going very fast and attacking.
Okay.
So their perception is altered. They are not recognizing a nestmate as a nestmate, and they're doing something they never do. They're biting.
Okay.
So we don't know exactly the mechanism, but it has to be messing with the perception of the colony odor and not allowing them to recognize someone as a nestmate, and also like speeding their alarm and aggressive reaction to levels where they first bite and then think.
Okay. Okay. Um, so like then we have received the question from David McCluskey. Do you understand why the hands are repelled by the fungus? Is it simply unpleasant to them, or is infection a threat to the hands?
Well, our data tell us that they always have to touch the material, so they are not avoiding it, not really.
Mhm.
Mm,
So they're
so,
always
so, so
they're
eh,
always touching somehow.
they need to touch it, and to show most of those weird behaviors, they had to bite it. So if they don't bite, and they don't bite for long, they are not gonna react like that. And something that I didn't show in the presentation because of lack of time is that these ants are not willing to walk on a strange material for a long distance. So the once the bird is able to trick the ants into allowing the attachment of material, I think they're good to go. They are not really looking forward into walking all the way down in the nest and entering the nest unless they perceive a threat.
Mhm.
So they're not repelled by just the smell or the presence of the fungus. They always interact with it.
Okay.
And
Yeah.
the
Okay.
colony, I think that allows for the colony to habituate to that material, just as they do with the metallic um, tags that we use. At the beginning, they attack and bite and bite and bite. At some point, they give up, and at some point, the tags acquire the smell of the colony, and they are ignored afterwards, after a while.
Mhm. So that we also have Bastormenta. So that I guess maybe you actually gave the information in the talk, maybe not. I'm not sure. How do the birds get the rhizomorphs?
Very interesting question. I also wondered the same. And when we started with the project, I thought it was a difficult material to find. But once you start seeing it, it's everywhere. It's everywhere. The rhizomorphs, it's crazy. There is a paper that demonstrates that the hyphae of the fungus lives inside the trunks or dead twigs. And when the resources are lacking, they start growing that structure that we call rhizomorph. Is a way of escaping without producing the fruiting body.
Mm-hmm.
And they can build, but it's more than that. So they grow the rhizomorphs, and these rhizomorphs build a web that traps the leaf leader. So it gets trapped in and entangled among the rhizomorphs, and they can start using the enzymes to digest the material. So the rhizomorphs are equivalent to a spider web. It's a trap for
Mm-hmm.
for leaf leader to be there. So there is variation probably depending on the genus of the rhizomorph that we're talking about. In the dry forest of Costa Rica, I've seen the rhizomorphs growing a lot from trunks, and some of those trunks are alive. They are not even dead. And I don't understand how or why that happens. But in Panama, the most of the rhizomers are those leaf traps and entangled among us. So to collect them, the easier thing to do is to look for, like, up in the canopy for those like bags of leaves, and
Mm-hmm.
there are tons of rhizomorphs in there. And one more thing that the technician of the electron microscope discovered is that it seems that the fungus has certain fluorescence in the UV um, wavelength. If the birds are able to see UV, they probably see rhizomorphs way easier than we do. It's
Okay.
not black for them, it probably shines.
So it is very unlikely actually that they don't get some, basically.
I think it's an abundant material. Once you find it and you know where to find it, it's very abundant.
So you never had to observe them trying to find an alternative because they were not finding it.
I don't know. Well, I don't have evidence for that because it's hard to guess what is the bird looking for, right? When
Yeah,
they're
yeah.
moving
Mm-hmm.
around. But there is evidence that in some other protected areas, the orioles, for example, mix grass or grassy leaves with the fungus, and then the proportion at which they use the fungus changes and decreases, and there's variability in
Mm-hmm.
there. Another dissertation to,
Okay, okay.
to work.
So like we have to stay updated to kind of know about this. Okay, so like we also receive a question from AB George. So first he said that your talk was fascinating. I had a question about the gaster curling. In Lasius, some work has shown that the ants curl their gaster to do acidopore grooming. Do these
Mhm.
ants also have an acidopore? Maybe
No,
what they do, sorry, you can reply to this first question and I can give the second part maybe.
well, quick answer, no, they don't have one.
Oh, they don't have. Okay, because like the comment that was coming after that, maybe what they do after the gaster curling could be an indication of what the purpose of the gaster curling is.
Yeah, that's true. So this is not a common behavior among ants. Some of them do. Some of the ants move the gaster upwards, not curling it under the thorax. And the few reports of ants doing that have to do with using substances to groom themselves.
Mm-hmm.
And the pseudomyrmex and that subfamily, Pseudomyrmecini, they have a stinger, say they don't have an aciopore. Um, and it's just that movement, and they don't do anything else with it. They don't use the first pair of legs, or they don't manipulate the abdomen in any way. It's just it's just doing that movement and then walking straight again.
Okay.
Yeah,
Okay.
hard to interpret. It's hard to interpret.
Yeah, okay, I understand. So we receive another question from Bastor Monta. How do the hens protect the bird nests that are so close to the wasp nest? I saw a documentary about wasps stinging chicks to death. So how do the bird hen and the hen guard survive?
Yes, and so that exactly, or that kind of thinking is what made me believe that the use of rhizomorphs is not directed only to the acacia ants, and that's why we were looking forward to test whether the alterations of behaviors would happen also with other organisms. And the wasps are also hymenopterans, and I would guess if these ants are affected, the wasps are affected too. They share a lot of the chemoreceptors, a lot of the physiology of the brain as well. Although before testing the wasps, we tested other ants, and these other ants are telling us no effect.
Okay.
And so now I'm torn, and I don't have an answer for the wasps. I can tell you that so we had to move down many of those yellow olive flycatcher nests that here in Panama are also sometimes close to wasp nests. And I thought it was going to be a disaster and very risky. And not really. If you don't move the branch too much, it's just a precise cut of the tip of the branch where the nest is. It will fall, and the wasps they don't get very altered.
Mm-hmm.
And the way they defend, I think, is because usually you have the wasp nest, and towards the distal end of the branch, you will have the bird nest. And the idea is that a mammal crawling on the branch will encounter first the wasps and then the bird.
Mm-hmm.
And so in principle, that's how it works. But I agree that the nests of the birds are very, very close to the wasps. And from the natural history observations that I have, it for me it's striking that even that I mentioned like more than 100 species of birds use rhizomorphs, very few build the entire nest out of the rhizomorph. And one of them is this one that nests either with wasps or with ants. So I don't think it's coincidence, but I don't have evidence, not yet. For the wasps, not yet.
Okay, interesting. So we have a question from Catherine Macri. When they don't recognize their nest mates, do you think maybe the substance of the rhizomorph impacts the cuticular hydrocarbons perception?
Yes, I think so. It is messing with either with the perception or with the interpretation of those chemicals in the brain. So it might be perceiving it, but interpreting them wrong. Now it's not recognizing them. And it's curious because if you had asked me, I would have thought that an ant that spent minutes and minutes interacting with the rhizomorphs and biting them would be the one that was going to be attacked by their nestmates because it changed the smell. Maybe it acquired some of the rhizomorph smell, but it's not like that. It's the other way around. And it seems like they are again very alarmed and in a hurry to check things. And it's not biting to death. Is they do the bite and they release, and they are not gonna bite again the nestmate, but I guess the poor nestmate is like, oh, come on, what's happening? Because that never happens.
Yeah, yeah.
But it's not they're gonna attack to death.
And is it something that already has been done, or that's something that you would like to do in the future to maybe, for instance, put some of the substance of the rhizomorph on hands and maybe after making electrophysiology, antennal electrophysiology or something, just in order to see whether a signal is completely altered or if it's the same thing, and then it means that maybe it is not a question of perception.
So the first thing that we were pursuing is to try to identify the chemical, and that one that I mentioned, whose name I cannot repeat,
Yeah.
that substance is one of the first candidate. And so yeah, maybe going into that direction, but not yet, because we weren't sure about what exactly is the component.
Mm-hmm.
And you can buy that synthesized and test it alone just to see if you can again recover the same behaviors in the ants.
Yeah.
And the extract in methanol recovered the grooming behavior a lot, but not the biting of nestmate behaviors. We couldn't recover that
Mm-hmm.
from our extracts. So I don't think we have the chemical in our profile. And it's a limitation of the method of
Yeah. Okay.
the substances that get extracted by like washing the material. But definitely that's one of the steps that should follow.
Okay. And so like we have time for a very last question from Tong. So since ants remove object regardless of toxicity, have you tested different concentration of chemicals?
Well, I'm not sure they remove things regardless of toxicity because they allow the rhizomorph to stay there.
Mm-hmm.
And that's the way in which one of the ways in which the flycatchers are able to build a nest with ants. That's the argument. So one of the reasons is that the material is very, very hard and it's hard to break or cut. And at the same time, every time they bite it, immediately they're grooming and biting and again grooming. So that ruins any chance of cutting the material. So I don't think I agree they remove the material regardless of toxicity.
Okay.
At least not the occasions. The other two ones, they seem to manipulate the material and put it away. But in that case, you see they use their mandibles as tweezers, like almost as tools and almost not squeezing the rhizome. So maybe it requires some mechanical forces to release whatever they are releasing. But
Okay.
this is very new, so I have more questions than answers. I'm sorry.
Yeah, so I guess that the take-home message also like stay tuned, people, like if you want more information about this.
Yeah.
So like we arrive to an end, but I can also say that we also receive a nice message like what an interesting talk. Thank you, Sabrina. Thank you for the great talk.